Hey everybody, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, just north of Philadelphia, where I've been teaching A&P since 2002. Today we begin the reproductive systems. This is going to be a long haul. We need to cover a lot of ground here, beginning with what exactly sexual reproduction is, since that's what we use to produce offspring and keep our species afloat in this universe. When we get down to basics, our existence is all about our ability to pass our genes on to another generation. All we have to do is live long enough to do that, and we've won the evolutionary contest. So buckle up for a handful of episodes to fully understand the human reproductive systems at the level you need for a career in healthcare. Before we get into the content, I'd love for you to join me for a conversation I had with a good friend, Dr. Kevin Patton. Kevin has done it all. He is a professor emeritus of A&P in Missouri, a past president of the Human Anatomy and Physiology Society, the author of several A&P textbooks from Elsevier Publishing, a past president of the Textbook and Academic Authors Association, and a professor at Northeast College of Health Sciences in their Masters of Science in A&P instruction program. Dr. Patton's contribution to anatomy and physiology education is immeasurable, and we are very fortunate to have gotten the opportunity to have him on to share his experiences and insight. So without further ado, please welcome Dr. Kevin Patton. All right, Dr. Kevin Patton, thank you so much for joining the show. I really appreciate you coming on.
Well, hi, Steve. It's good to be here. I always enjoy chatting with you. Usually it's at our Human Anatomy Physiology Society meetings that we run into each other. So it's kind of fun to do a one-on-one right now.
Absolutely. I mean, HAPS is a great organization. I talked about HAPS on the show a few times. The Human Anatomy and Physiology Society is just such a wonderful confluence of A&P instructors, and we all kind of know each other. It's like the same people every year for the most part. And I really, really love that community that we've gotten with that. And I know that you played a major role in that, and we'll talk about that later. But I want to get the listeners a little feel, even though they already heard about you from the intro, a little feel about your background, where you've taught anatomy and physiology. Have you taught anything else other than that? Things like that.
Well, yeah, as far as my teaching goes, I actually started out teaching high school. And I taught general bio and anatomy and physiology in high school for a couple of years, and had an opportunity to teach at a private two-year college. And I just loved it. And I think a lot of it has to do with the fact that my mentor, the person who had been there forever, we shared an office, and she just really shared everything with me. And she just really took me under her wing, and I really learned from a master of teaching. And so that's how I got into it. And then I got into a community college in St. Charles, Missouri, that's near St. Louis. And the community college had just started, so I was one of the founding faculty members. I was there for 26 years full-time, and then for a number of years part-time after that. At the same time, kind of overlapping that, before I retired from full-time teaching, I started in a program at Northeast College of Health Sciences. That's in upstate New York, the Finger Lakes area. And they were starting an online master's program for teaching or training people who either were or want to start teaching anatomy and physiology, either at high school, college, even professional like medical school, chiropractic school, physical therapy, dental, whatever. They actually connected with HAPS and start looking around to see, well, who are some people who'd be good mentors for people? And they picked me as one of them. And I mean, at first they were just asking me for some ideas, what my input was. And they said, hey, you want to be on our faculty? And so I did that. And that was one of the best moves I ever made, because it was late enough in my career of teaching A&P that I really felt like I did have some things to offer, that I had made some mistakes and I learned from them. And I was glad to share those insights with other people. We call it the HAPPY program, Human Anatomy and Physiology Instruction. So in the HAPPY program, we have our fellow faculty, and we also have, the cohort of learners, some of whom have already been in the classroom, some of whom are yet to face their first class full of students. and we make those connections and we network. And that's really what grounds all of us is that interaction and that network and so on. So what I try to do when I'm teaching and mentoring other A&P faculty is like take yourself back to when you were a student and think about how you didn't do it by yourself. There's always people around you to help you and so on, and take advantage of that. So
That's really neat. I love hearing about that HAPPY program up at the Northeast College of Health Sciences, which is my alma mater, by the way.
I knew that.
Yeah.
Yeah.
And I'm one of those people who started teaching A&P at night while I was practicing and really, really loved it. They gave me more and more sections, and then eventually they hired me full time, and I left everything else behind. And I think that this program is good for making sure that people who want to teach A&P, they get trained correctly and they're respected for the training.
What I like about it is that there are two different things that we emphasize, and we integrate them together as much as we can. So half the program basically is a review of all the major topics that are taught in an A&P course. So of course there are some people who would be really strong in like skeletal-muscular stuff, and they really don't need much of a brush up, but they need some practice in, well, how do you pick those topics, for an undergraduate student? And how do you take those topics and really make it clear enough for someone who's never seen or heard of this before, or at least not much? But then they get, maybe a little bit further along, and now here we are in the endocrine system, and they may not remember as much about the endocrine system from their early training, or maybe they never got that much. And so now here's an opportunity to, really dive in and brush up on that. So by the time they're done with our program, they're really strong in all the major areas of human anatomy and physiology. But all along, they're not only taking some courses in how to teach and what are the latest evidence-based strategies in teaching. They're doing that, but as they're learning, like I was doing the nervous system course, so we brushed up on all the aspects of the nervous system, which is a pretty big and complicated system, right? But while we're doing that, we're also learning, well, how would you teach the nervous system? How would you teach about the different regions of the brain and what they do and how they interact? How would you approach all of those cranial nerves? How would you approach all those spinal nerves and their branches and what they do? I mean, you can't learn everything about the nervous system, but what are the main ideas, and how would you approach that with your students, and how would you, where are the pain points that students are going to run into that you can help them with? And so they practice, how to teach it, and how to, what makes a good, authentic test for the, each of those subjects and so on. So it's, I think, a really great approach to training people for the, teaching human anatomy and physiology.
As a college instructor, I think that what people might not realize is that most people who teach in college, instructors, professors, they're subject matter experts. They do not have any classical education training. We don't have education degrees, and that makes it so that it's a little challenging in terms of learning how to teach the topic, even though we're not trying to teach first graders how to read. We're teaching adults. But still, it is a challenge. I tell my students all the time that my job is to simplify the content without being inaccurate. I do want to switch gears a little bit and ask about your podcast, because like me, you have a podcast, and I think you've had it for a little bit longer than I have, and it's really, really good. It is a podcast that is mostly geared toward people teaching A&P, which is so appropriate based on what you do, and it's called The A&P Professor. So could you tell me a little bit about why you decided to start a podcast, and tell us about your podcast, where people can find it, all that good stuff?
Yeah, sure. Well, the podcast you can find anywhere you listen to audio. Just search for the A&P Professor, or search for my name, Kevin Patton. Sometimes that works. It depends on where you're at when you're searching, but that's one way to get to it. Or you can go to theapprofessor.org and find it there. So that's theapprofessor.org, and And it's, if you have a podcast app or something, it should be in there. And you can, by the way, go to the App Store on your device, whether it's Apple or Android or whatever, and just search for the A&P Professor in the App Store. And we have our own app, so you can listen to it in that app.
I will put links to all of those things in the show notes for everybody. So, all of the different ways that you can get access to that podcast.
The reason I started doing it is, I mean, I've always had a love for doing audio stuff. My very first professional paid job where I wasn't working in my dad's print shop and an actual outside employer was at the St. Louis Zoo. And my job was to sit on the back of the little train they have that goes around the zoo and give the tour and talk about the different animals and other things that people saw. And I loved that. So, they never saw me because I was way at the back of the train. I'd come around and take tickets, and they'd say something like, "Hey, can we talk to that guy who's doing the narrations?" Like, "Hey, that's me." But anyway, so I always enjoyed the audio stuff. And when I was in college, I took a course in broadcasting and learned how to do radio type stuff. So I just had a love for that, but that's not the direction I went in. But I always had that in the back of my mind, audio stuff, and how much I enjoyed doing audio. And when I got to the end of my teaching career, teaching undergraduate anatomy and physiology, and was getting ready to start, teaching, mentoring people who, wanted to be A&P teachers or already were at Northeast. I thought, a lot of schools, especially big universities, they have this thing that professors do when they retire called their last lecture. So it's kind of a thing where they invite anybody who wants to come in the college or university community, whether it's students, faculty, alumni, community members, whatever. And that professor gives his or her last lecture on any topic. And so it's like, what have I learned in this career of teaching and research and doing other things? And so I felt like I did when I started teaching at Northeast. I felt like I had some things to share with other anatomy and physiology professors. So that's kind of that's where the inkling came from and why audio. That was it. And that was at a time when audio was, or podcasts especially, were get very rapidly becoming popular, and people were actually listening to them. Before that, it was kind of a little niche thing where a lot of people, not very many people listen. So it seems like just a whole bunch of different things came together.
Well, I think it's really great. And I think that students would be surprised at how much most of us do to try to get better at what we do. Whether it's going to HAPS conferences or listening to a podcast like yours so that we can get as much help and tips and tricks and things like that from all of our colleagues to just make us better at what we do. So, in addition to your podcast, I also know that you are the author of a very popular series of A&P texts as well, but I didn't know that you were also the president of the Textbook and Academic Authors Association. So I would love for you to share a little bit about the textbooks that you author, and then maybe some experience on the Academic Authors Association. Is that like a union of textbook authors?
Yeah, it's I can start with that end of it. The Textbook and Academic Authors Association is a group. It's not a union in the technical sense of we don't, bargain collectively or anything like that. The textbook industry is its own thing. There's a lot of aspects of it that most people really don't have a clue about until they get into it, and then they're like, "Oh gosh, I didn't know it worked that way." Um, and so, you have a lot of authors who, maybe, well, not maybe, they will have taught. I mean, that's how textbook authors are recruited to do that. Is that they either have an idea for a textbook because they've been teaching and they realize that, they're the textbooks that they've used just don't serve them that well, and they want to write their own. And so, I mean, if they're really passionate about it and really believe they can do it, then they'll approach a publisher and they'll see if the publisher will publish it. But very often, especially more recently, it's the publisher who's looking for authors to either write a new textbook or maybe the authors of that textbook are retiring, so they need new people. But there's a whole bunch of other skills that you need to learn, and you need to learn them quickly. So TAA is sort of the HAPS for textbook authors. And when we get together for our conferences, it's like a conference of lighthouse keepers. And we get to share our stories with one another, share our tips, share our shortcuts, relationships with publishers. So it's all about making the writing of textbooks better in a variety of different ways and helping us do that job of being a textbook author. And my textbooks in human anatomy and physiology, they're geared toward different kinds of A&P courses. There are those that are 2 semester A&P courses. That's probably the most widely taught kind of undergraduate A&P course. But there are also 1 semester courses. So we have a couple of different versions for those. One of them has a little extra disease pathophysiology content in it, and the other one is more of a basic structure and function of the body type approach. Our publisher is Elsevier. You know, one of the biggest frustrations for me is we're always doing revising of the books. And I don't know, I think a lot of people get the idea that we just slap a new cover on it and maybe change out a couple of the pictures inside, and that's the new edition. There are literally thousands of changes that we make on our books when we revise them. And I know it's not just me. Just in the manuscript, our document had a way to count up the number of changes that were made. It was like 2,500 or something changes we made. And all of my friends who were A&P textbook authors, they responded back and everybody was doing the same sort of thing. So it's a continuous improvement process because, we've learned some new things in science, but also, I've learned new things on how to write more clearly and maybe some better examples of things and, all of that.
And there's some things that have changed more recently. A lot of these textbooks are print on demand now. So instead of an edition every 3 or 4 years, the publishers are doing annual updates. A lot of times my students will say, well, the human body hasn't changed in 200 to 300,000 years. Why are the textbooks changing? It's because we learn more every year. So you mentioned HAPS a few times. I do want to bring HAPS up one more time. That's the Human Anatomy and Physiology Society. What I didn't realize is that you were involved very early on. I got involved in 2009. That was my first National HAPS conference in Baltimore. I'd like to know your involvement in the beginning of the Human Anatomy and Physiology Society.
Well, I was involved early on. I wasn't at the very beginning. The organization started because a guy named Bob Anthony, he was teaching at Triton College up near Chicago, a community college up there. He's teaching A&P, and I think there was an informal group of A&P instructors in that area that would do things together every once in a while. So he organized several days of workshops at his place and invited people not only from the area but from all over the place. And I have some friends from Missouri and from Oklahoma and that were at those original workshops that Bob Anthony did. And they said, not everybody can make it to Chicago. Let's start moving these around. Let's make this more of a national thing. And so that was the seed of HAPS. And they had their first non-Chicago meeting in Reno in 1989, and I couldn't go to that, but I joined the society then. In 1990, was in Madison, Wisconsin. That was my first live one. And I remember at our business meeting, we all voted to become real. That is to incorporate as an actual organization and really commit to not only having conferences every year, but doing, starting a program that's now huge of all kinds of different services and ways to improve anatomy and physiology teaching, not only in the United States, but throughout the world. Right there in 1990, my first meeting, they were encouraging me to be a host in St. Louis, which is, my home base. We'll never forget Gary Johnson, another early HAPS member. He was the host. And so I went up to him and I said, Gary, I said, so tell me, what's it like? I mean, is this like an impossible task? Because back in those days, we didn't have any help. If you were the conference coordinator, it was all on you. I mean, you could get your friends to help you, but we didn't have a professional staff the way we do now. That not that the conference host job is easy, but it's a lot easier now than it was back then. So I was asking Gary, and he said it is a wonderful feeling to have done one of these. And so that kind of nudged me forward. In 1995, I hosted one in St. Louis, and everything that could go wrong did. But you know what? It's HAPS, and we're all buddies, and we all kind of pulled together and made it work. In the end, it came off pretty good. A lot of those things that went wrong, many people didn't even realize they went wrong. It was behind the scenes and so on. And once you're a conference coordinator, everybody knows you. And so I kind of went through the cycle of being an officer. So I was secretary treasurer, which back in those days was a combined position, and then became president. While I was president, we decided to have another position called president emeritus. They could continue to be sort of an ambassador, perhaps, and also be available to be tapped for special projects. And I got tapped for developing what we call HAPS Institute, which is a program of professional development within HAPS, where it's sort of like, I don't know, HAPS on steroids in a way. It's like specific courses. They're not, a short-term conference or workshop, but they're more of a, something lasting several weeks and really focused. Many of them also carry graduate college credit with them, so they really are literally courses. So, and that was fun. That was a lot of work, but it was fun. And by the way, when I did my conference in 95 in St. Louis, I ran into Gary Johnson. And I said, Gary, you said this was great. And he said, no, you didn't listen to my words. It's a great feeling to have done it. You're going to feel great about this in a few weeks, but right now you're going crazy. I know, because I did the same thing. And he was right. And it was a great thing to have done. And all the service I did in HAPS, even when the going got tough, I'm so glad I did it. Not only for the effects that these things continue to have, but it really helped me a lot, just personally. I think anytime that we can step up and do service for our peers, that we get as much, if not more, out of it than the folks that we're helping to serve.
Well, that's great. Well, I can't thank you enough because HAPS has been an enormous influence on me as an instructor and an author, and I never miss. I can't wait till it comes back around. I'm always sad when I leave the national conference. Well, Kevin, thank you so much. You've been really generous with your time. So glad that you were able to come on and we finally got to talk on the podcast. I really appreciate you coming out. Is there anything else that you want to share?
No, I have lots of stories up my sleeve. If anybody wants to listen to my podcast, I tell some of those stories. And as I mentioned before, it's directed more for A&P faculty, but I think A&P students might enjoy listening to some of the stories I tell.
I agree. I think that any student listening to this podcast will enjoy your podcast as well, just to pull back the curtain and see what it's like on the instructor side as well. And I think getting a whole like bird's eye view of A&P, I think could do nothing but be beneficial.
Boy, Steve, I really appreciate it. It's always fun to chat with you. And you know me, it's get me started on some of these topics like A&P teaching, and it's like hard to get me to stop. So I really appreciate the opportunity to do this.
Well, I really appreciate you coming on and sharing with the students, and I will see you in Kansas City.
Sounds great.
Thanks, Kevin. That was great. I am a huge fan of Kevin's. He has done so much to advance the field of anatomy and physiology education. Everyone who studies A&P and or teaches it owes Kevin Patton a huge debt of gratitude. I can't thank him enough for joining me on the show. I have posted links to Kevin's website, his podcast, the Northeast College of Health Sciences Master's Degree Program for Teaching A&P, and the website for the Human Anatomy and Physiology Society, HAPS, in case you're interested in this outstanding professional society. Check them out. Incredible stuff. Having said that, it's time to begin our discussions of the reproductive systems.
Bombas makes the most comfortable socks, underwear, and T-shirts.
Warning, Bombas are so absurdly comfortable you may throw out all your other clothes.
Sorry, do we legally have to say that?
No, this is just how I talk, and I really love my Bombas.
They do feel that good. And they do good, too. One item purchased equals one item donated.
To feel good and do good, go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
We'll start by talking about how we pass our genes on to our offspring. As mammals, we utilize sexual reproduction. This means two biological parents contribute genes to the offspring, so every child is not genetically identical to each other or either parent. We do this by producing gametes, or sex cells. The gametes are a spermatozoon, or sperm, and an ovum, or egg. The sperm and egg meet and combine their genes to form a zygote, which is a fertilized egg. Fertilization is also referred to as conception, so often you'll hear the term conceptus used to identify the resulting cells from fertilization. The first 8 weeks post-fertilization is the embryonic period. After that, the conceptus is considered a fetus until birth. Way back in episode 7, I talked about chromosomes. The number of different chromosomes a species has is called its ploidy, Ploidy. Humans have 23 different chromosomes, and most somatic cells have a pair of each, totaling 46. The biological mother and father each get to contribute their version of each of those 23 chromosomes. When a cell's chromosomes are paired like that, we call that cell diploid, because it has 2 of each chromosome, Di for 2. The chromosomes in gametes are unpaired, making them haploid, meaning that they only have 23 total. This is how zygotes get a pair of each. The sperm and the ovum combine their 23 each into one diploid nucleus of the zygote. If zygote formation is going to be successful, 2 requirements must be met. First, the gametes must be able to move so they can meet and contact each other. The second is enough cytoplasm and nutrients for multiple cells must be available. After the egg cell is fertilized, the zygote divides into the first cells of a developing embryo. Without the organelles and intracellular nutrients to sustain it for a while, it won't survive. Eventually, the embryo will be supported by an outside source, but until then, its cells must take care of themselves. These 2 requirements are met separately by our 2 different gametes. The ovum contains the cytoplasm and nutrients necessary for multiple cells, and the sperm has a tail-like flagellum that propels it along so it can move toward the ovum. The formation of these gametes is called gametogenesis, and while sperm production and oviproduction differ in many ways, they both utilize a version of nuclear division called meiosis. In episode 7, I discussed mitosis, which is the nuclear division of somatic cells that result in 2 identical diploid daughter cells. Meiosis is nuclear division of sex cells, and that results in 2 or 4 non-identical haploid gametes. So let's talk about meiosis in general before we move on with sex and gender. I'll cover the specifics of spermatogenesis and oogenesis, which is oviproduction, in later episodes. One of the key objectives of meiosis is to ensure genetic diversity in the offspring. This is achieved by shuffling the genes of the germ cells so that each chromosome in every gamete carries new gene combinations that didn't exist in the parent cell's chromosomes. Meiosis includes 2 rounds of nuclear division, meiosis 1 and meiosis 2. We use Roman numerals to represent the 1 and 2. Like mitosis, each round of meiosis is divided into 4 stages: prophase, metaphase, anaphase, and telophase. During meiosis 1, we add a 1 to each phase, prophase 1, metaphase 1, etc. During meiosis 2, we add a 2 to each phase, prophase 2, metaphase 2, etc. Spermatogenesis, the production of sperm cells, occurs in the seminiferous tubules. It involves the transformation of a relatively large diploid germ cell into 4 small, mobile, haploid cells with flagella. Biological males continue to produce sperm throughout their lives. On the other hand, oogenesis, the production of egg cells, or ova, takes place in the ovary. Biological females are born with all the eggs they will ever have. During oogenesis, the process of meiosis is halted during the second round until fertilization occurs, if it ever does. It's important to note that instead of producing 4 gametes, the outcome of oogenesis is 1 fertilized egg and polar bodies that degenerate. In humans, each somatic cell contains 23 pairs of chromosomes, resulting in a total of 46 chromosomes. These chromosomes carry different genes that code for various traits. We inherit one version of each chromosome from each parent. In this context, the term homologous means having the same position or structure. For example, chromosome number 1 you get from your biological mother, and the chromosome number 1 you get from your biological father are homologous to each other. Or we can call them homologs. Our diploid cells have 23 pairs of homologous chromosomes. The diploid parent cell that produces gametes is called a germ cell. Before meiosis I, DNA replication occurs in the germ cell, resulting in the chromosome having 2 copies of its DNA. However, DNA replication does not occur again before meiosis II. The first phase of meiosis I is prophase I. During prophase I, the chromosomes condense and shorten, similar to the prophase of mitosis. However, in prophase I, homologous pairs of chromosomes come together side by side to form tetrads of chromatids. The prefix tetra refers to 4, as each tetrad consists of 4 chromatids. This process is called synapsis. Also, like in mitosis, the nuclear membrane begins to break down. During prophase I, a crucial process called crossing over takes place. While the homologous chromosomes are very close to each other, the DNA strands of each break in the same genetic location. The broken ends cross over to the other homolog and attach to its DNA. This results in the swapping of several genes between homologous chromosomes. This genetic exchange occurs between the chromatids of homologous chromosomes, creating new gene combinations that didn't exist in the parent cell. The process of crossing over is unique to prophase I. It doesn't happen in prophase II, and it doesn't happen in any other phase, and it doesn't happen in mitosis. And it contributes to genetic recombination. The next phase is metaphase I, where the homologs line up 2 by 2 along the metaphase plate. In anaphase I, each member of the homologous pair crawls along the spindles of microtubules toward opposite poles, resulting in 23 chromosomes, each carrying 2 genetically different chromatids on each side of the cell. Why are they genetically different now? Because we crossed them over in prophase I and swapped our genes with each other. Telophase I is the end of meiosis I. A nuclear envelope reforms around each group of the 23 chromosomes. A cleavage furrow splits the cell in 2, resulting in the division of the cytoplasm and the formation of 2 genetically distinct haploid cells. These haploid cells will each undergo another round of division called meiosis II, which closely resembles the mitosis that takes place in somatic cells. But there is no DNA replication before meiosis II. So now what we've got are 2 haploid cells. They each only have 23 chromosomes. However, each of those chromosomes still has 2 chromatids. And each of those chromatids, while not identical to each other, have all the DNA required for that particular chromosome. That's very important as we get into meiosis II. Okay. Prophase II begins with the breakdown of the nuclear envelope again. Remember, the chromosomes each consist of 2 non-identical, genetically distinct chromatids. Metaphase II sees these chromosomes lining up at the metaphase plate, single file, as the homologous pairs are now separated into different cells. During anaphase II, the chromatids separate at the kinetochore of the centromere and migrate to opposite poles of the cell. This is like the anaphase of mitosis. Finally, in telophase 2, the cell undergoes cleavage, and the nuclear membrane reforms again. The result is 4 haploid cells, each containing 23 chromosomes, and are genetically distinct from the parent cell. These cells will be referred to as gametes. Due to the 2 rounds of division in meiosis, each male germ cell produces 4 haploid, genetically different gametes. Again, in females, the result is 1 fertilized egg and polar bodies that degenerate. There is still more development that has to take place before these haploid cells become sperm or ova. But in general, this is how it starts, with meiosis. Okay, so now that we know where these gametes are coming from, let's speak simply and biologically for a minute before we get into the existing nuances that are not so simple. In any sexually reproducing species, by definition, an individual that produces eggs is female, and the one that produces sperm is male. It's not always that simple, and we'll discuss that later on. I'll talk about chromosomes in a bit, but genetically speaking, the sex of a person with a Y sex chromosome is classified as biologically male, and one without a Y is classified as biologically female. Now, sex is not the same as gender, and we'll get into that later. But to keep things simple, when I say male and female, I'm talking about biologically male and female. Sex, not gender. Keep that in mind as we move forward. In order to deliver the sperm to the female reproductive tract, the male must have an organ of copulation, a penis. As I mentioned in episode 55, which covers the urinary system, the urethra is a tube running through the length of the penis. The urethra is utilized by both the urinary system and the reproductive system, as it is also the conduit through which semen is ejected. Semen is the fluid medium used to deliver sperm. The urethra is strictly urinary in females. The female must also have a copulatory organ for receiving the sperm, and that's the vagina. The vagina is also used to give birth to the offspring when the time comes. As you're likely aware, these organs are the most obvious differences between the sexes. The fertilization of the egg takes place within the female reproductive tract, and that's where the offspring develops. This puts the burden on the female for supporting and nourishing the developing offspring, which we call an embryo and fetus, depending on how far along the development is. All right, before we move on, I want to define the term natal. N-A-T-A-L. Natal pertains to the time of one's birth, so prenatal refers to events taking place in the expectant mother's body before birth, and postnatal refers to those after. The neonatal period is the time early in the life of a newborn infant. Okay, so let's talk about the reproductive system in general before we discuss the specific differences between males and females. Reproductive anatomy is divided into primary and secondary sex organs. The primary sex organs produce the gametes. Generically, they're called gonads. In the male, they are testes, which is the plural of testis. For female, it's the ovaries, which is the plural of ovary. Try not to get sucked into using the word testy for one testis. It's actually testis for one, testes for two. The secondary sex organs are non-gonadal organs, but are still necessary for reproduction. In the male, they are the structures associated with storing, supporting, and delivering sperm. They include ducts that carry sperm from the testes, the glands that contribute to semen, and the delivery organ itself, the penis. In the female, they are the tubes the ovum travels through after being released by an ovary, the structure that supports and nourishes the embryo and fetus, and the vagina, which I mentioned earlier. Some reproductive organs are within the pelvic cavity. These are classified as internal reproductive organs. The exceptions to this are the testes and the beginnings of the ducts that carry sperm toward the urethra, which are classified as internal but are within the scrotum, not the pelvic cavity. Some reproductive organs are outside the pelvic cavity, and these are classified as external reproductive organs. The external region of the body inferior to the pelvic cavity is called the perineum. This is where the external reproductive organs can be found. We also exhibit secondary sex characteristics, which are features that further distinguish male from female and play a role in how we attract a mate. In the animal kingdom, we see mate attraction all the time. Male and female birds may be different colors, body size, have different dances, etc. They typically appear only as an animal approaches sexual maturity. In humans, we attract mates differently and usually determined culturally. Also, what makes someone attractive to a mate is fairly subjective and, quite honestly, manipulated by society, advertisers, marketers, etc. However, generally speaking, pubic and axillary hair, their associated scent glands, and the pitch of the voice are physical characteristics that signify sexual maturity. These typically arise during the teenage years in humans. There are other traits commonly considered male secondary sex characteristics as well. For example, facial hair, visible hair on the torso and limbs, and a relatively muscular physique. In females, these secondary sex characteristics include the distribution of body fat, enlargement of the breasts, which is independent of lactation, milk production, and finer, less conspicuous body hair. Genetically, whether a zygote develops into a biological male or female is determined by the sex chromosomes, which are chromosome number 23. Chromosomes 1 through 22 are called autosomes, but 23 is called a sex chromosome. A sex chromosome can be either X or Y. That's how we indicate which one we're looking at. Every egg contains an X chromosome, so that is all the female can contribute. But a sperm can carry an X or a Y, and the split is 50-50. If a sperm with an X chromosome fertilizes an egg, the result is a zygote with an XX for its sex chromosomes, and that usually develops into a female. If a Y-bearing sperm fertilizes an egg, we get an XY zygote, which usually develops into a male. So the offspring sex is typically determined at fertilization. Furthermore, it's the sperm that determines a sex because it's the gamete that can be either X or Y. So now think about Henry VIII, King of England, back in the 16th century. Throughout his adult life, he married 6 women. Most of those marriages ended in annulment, divorce, or execution. Why? Well, a lot of historians say it was because he blamed them for never having a son to be his heir to the throne. Well, they didn't know much about genetics back then, but now we know that whether a baby is male or female is determined by the sperm, not the egg. So of course it was no one's fault because it's all up to a 50-50 chance each time, but if he insisted on blaming someone, it should have been himself. The differentiation between the sexes goes beyond the genetic determination from X and Y chromosomes. Prenatal hormones secreted by the expectant mother also play a role as they interact with the genes of the fetus, which is what the conceptus develops into about 8 weeks after fertilization. You can review episodes 33 and 34, which cover the intro to the endocrine system and how hormones affect their target cells. The main thing to remember is that what makes a cell a target for a hormone is that it has receptors for that hormone.
Bombas makes the most comfortable socks, underwear, and T-shirts.
Warning, Bombas are so absurdly comfortable you may throw out all your other clothes.
Sorry, do we legally have to say that?
No, this is just how I talk, and I really love my Bombas.
They do feel that good. And they do good, too. One item purchased equals one item donated.
To feel good and do good, go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.
Despite the sex chromosomes of the zygote, an embryo is undifferentiated when it comes to biological sex. Its gonads begin developing into gonadal ridges about 5 to 6 weeks post-fertilization. Each gonadal ridge is found next to 2 ducts. the mesonephric duct, and the paramesonephric duct. The reason they have neph in their name is that the mesonephric duct serves as the mesonephros, a primitive kidney-like structure that does not develop into the kidneys in humans, even though it does in other animals, like amphibians and fish. But it does have some kidney-like function. In human males, the mesonephric ducts develop into the reproductive tract, and the paramesonephric ducts degenerate. In human females, it's the opposite. The paramesonephric ducts develop into the female reproductive tract, and the mesonephric ducts degenerate. That seems simple enough, but the key is, how does the embryo know which of these should occur? Well, the Y chromosome has a gene called SRY, which stands for sex-determining region of the Y. The SRY gene codes for a protein called testis determining factor, or TDF. What do I mean by code for? Well, go back and listen to episode 6 on DNA structure and protein synthesis. See how all this builds on previous knowledge? Okay, so what does testis determining factor do? Well, TDF interacts with genes on other chromosomes, including one on the X chromosome that codes for the synthesis of androgen receptors, as well as other genes that initiate the development of male anatomy. Recall that androgens are the steroid hormones that are responsible for male secondary sex characteristics. So if this embryo has XY sex chromosomes, TDF gets the ball rolling on the embryo developing into a male. By the 8th or 9th week of development, the male gonadal ridges develop into a primitive testis that begins to secrete testosterone. Testosterone stimulates the ipsilateral mesonephric duct to develop into the ducts that carry sperm from the testis to the urethra. The testis also secretes a hormone called Mullerian inhibiting factor, MIF, which stimulates the ipsilateral paramesonephric duct to degenerate so it doesn't develop into the female reproductive tract. Why is it called Mullerian inhibiting factor? Well, the paramesonephric ducts used to be known as the Mullerian ducts, but eponyms, names based on people, are being phased out in anatomy, so hopefully we'll stop using that. Interestingly, adult males still have a tiny Y-shaped remnant of the paramesonephric ducts near the prostatic urethra. It's like we have a tiny set of uterus and uterine tubes in there. Okay, so that's how a male develops. Androgens stimulate the development of male reproductive ducts and inhibit the development of female ducts. If the sex chromosomes of the conceptus are XX, there is no SRY gene to code for testis determining factor. So in the absence of androgens and their receptors, the mesonephric ducts are not stimulated to develop into male ducts, and the paramesonephric ducts are not inhibited, so they can develop into the female ducts. So what happens if there is a disruption in this process? There is a condition called androgen insensitivity syndrome, or AIS. What happens is that a child who had been growing up as a girl starts going through puberty and the typical female changes begin to arise. However, she doesn't begin to have her period or menstruate. Upon examination, doctors end up finding testes in the abdomen instead of ovaries. And genetic testing shows the sex chromosomes XY, which is genetically male. The testes produce testosterone at normal male levels, but the gene that codes for androgen receptors is faulty. And the target cells are unaffected by the testosterone because they don't have working receptors. So even though testes develop on the inside, the typical masculinizing effects do not show up on the outside. The external genitals are female. Testes produce a small amount of estrogen, and without testosterone receptors, the effect of estrogen can't be overridden. So at puberty, breasts and other secondary sex characteristics develop. There are no ovaries, vagina, or uterus. Thus, such individuals are male with respect to genes and gonads, but not with respect to genitals. Since undescended testes present a high risk of testicular cancer, the testes should be removed when they're discovered. AIS occurs in up to 1 person per 20,000. That means that there's about 400,000 people in the world that fall into this category. Now let's discuss the difference between sex, intersex, and gender. About 99% of individuals fall into the categories of either male or female as I've been describing them. The term 3G male and 3G female is one way to put it. The G's stand for gonads, genes, and genitals. So 3G males have all three male: testes, XY chromosome and SRY gene, and male genitals. They've got the gonads, the genes, and the genitals. 3G females have ovaries, XX chromosomes and no SRY gene, and female genitals. So they've got all three Gs as well. That's 99%. But not everyone fits into this binary classification. About 1% of people are intersexual, meaning that there is some level of disparity among the three Gs. Intersex means that not all a person's Gs are male or female. They have some combination of male and female when it comes to gonads, genes, and genitals. Androgen insensitivity syndrome is one example, which I've already talked about. Another scenario that happens results when the meiotic crossing over between regions of the X and Y chromosomes allows the male-determining SRY gene to transfer from a Y to an X chromosome during spermatogenesis. That means a sperm with that modified X chromosome can pass the male SRY gene to an XX child. That means a chromosomal female will produce the testis determining factor and develop male genitals. So we have a boy with female chromosomes. Or you can say we have a girl with a male sex determining gene and male genitals. Another way intersexuality can occur is for an XX fetus to be masculinized by abnormally high prenatal androgen exposure and be born with male-like or ambiguous genitalia. This is called adrenogenital syndrome. These individuals have female genes and gonads, but male genitalia. Since genitals are typically all that are checked at birth, they are likely misidentified as boys when they're born and raised as boys by their parents. For these and other reasons, we tend to see a disconnect between the genes, gonads, and genitals in about 1% of all people. That's about 80 million people worldwide. That's more than the entire population of the United Kingdom, Thailand, or France. Now, gender is not the same as biological sex. The World Health Organization defines gender as referring to the characteristics of women, men, girls, and boys that are socially constructed. This includes norms, behaviors, and roles associated with being a woman, man, girl, or boy, as well as relationships with each other. As a social construct, gender varies from society to society and can change over time. That basically means gender is a person's psychological sense of masculine, feminine, or other sexual identity that can differ from that of a person's biological sex. So while it would be very simple if we did, humans don't fall into two binary categories. For many biological reasons, gender is a spectrum from feminine to masculine in terms of anatomy, physiology, and psychology. Gender identity is one's innermost concept of self as male, female, a blend of both, or neither. Essentially, how individuals perceive themselves and what they call themselves. One's gender identity can be the same or different from the sex assigned to them at birth. I'm sure at this point you've heard the term transgender. Transgender is an umbrella term for people whose gender identity and/or expression is different from cultural expectations based on the sex that they were assigned at birth. Okay, let's finish this episode with the prenatal development of the external genitalia. Embryonically, whether male or female, the genitals develop from identical structures. 6 weeks post-fertilization, the embryo displays 3 distinct precursors to the genitals. The genital tubercle, a bud found anteriorly in the median line, urogenital folds, 2 bilateral medial tissue folds slightly posterior to the genital tubercle, and labioscrotal folds, a larger pair of bilateral tissue folds lateral to the urogenital folds. It's not until the 9th week post-fertilization that the fetus begins to show sexual differentiation. And it's not until week 12 that we can distinguish between male or female genitalia. We haven't discussed the reproductive anatomy yet, so bear with me as I assume you know the basics. In the female, the genital tubercle develops into the clitoral glands. This is the head of the clitoris. The urogenital folds develop into the labia minora, the smaller inner lips of the female genitals, and the labioscrotal folds develop into the labia majora, the larger outer lips of the genitals. In the male, the genital tubercle elongates to form part of the penis. The urogenital folds fuse, enclosing the urethra to complete the formation of the penis, and the labioscrotal folds fuse to form the scrotum. Since male and female organs develop from the same embryonic structures, we use the term homologous to describe specific organs coming from common, specific embryonic structures. For example, the penis is homologous to the clitoris, and the scrotum is homologous to the labia majora. Testes and ovaries initially develop in the superior aspect of the abdominal cavity, and eventually they descend into their final destinations: the pelvic cavity for ovaries or the scrotum for testes. In the embryo, a connective tissue cord called the gubernaculum anchors the gonads to the floor of the abdominopelvic cavity. As the male gubernaculum continues to grow, it passes between the internal and external abdominal oblique muscles and into the scrotal swelling. Independently of migration of the testis, the peritoneum also develops a fold that extends into the scrotum as the vaginal process. The gubernaculum and vaginal process create a canal that passes anterior to the pubic symphysis. This is called the inguinal canal. In males, the inguinal canal has an opening in the pelvic cavity and the scrotum. Sometimes a part of the small intestine protrudes into and gets trapped in the inguinal canal, resulting in an inguinal hernia. If you're a biological male, you may have experienced a physical exam in which the practitioner puts one or two fingers on either side of the base of the scrotum and asks you to cough. The coughing increases abdominal cavity pressure, and if there is an inguinal hernia, the practitioner will feel the intestine being pushed into the inguinal canal against their finger. What they should feel is nothing, but they're checking for inguinal hernia. The testes begin to descend around week 6 post-fertilization. The superior part of the embryonic gonad degenerates and its inferior part migrates inferiorly, guided by that gubernaculum. It's not until the 7th month that each testis passes through its ipsilateral inguinal canal and into the scrotum. Testicular arteries and veins, lymphatic vessels, nerves, spermatic ducts, and extensions of the internal oblique muscle continue to elongate and follow the testes through the inguinal canal. The vaginal process becomes separated from the peritoneal cavity and persists as a sac called the tunica vaginalis, which enfolds the anterior and lateral sides of the testis and protects it from abrasion against the inside wall of the scrotum. About 3% of boys are born with undescended testes, which is called cryptorchidism. Most of the time, they make their way into the scrotum before the infant's first birthday. If not, a testosterone injection or a simple surgery can help them along. The reason the testes are suspended from the body in the scrotum instead of being in the pelvic cavity, like the ovaries, is that the optimal temperature for spermatogenesis is 35 degrees Celsius, while the core body temperature is 37 degrees Celsius. So uncorrected cryptorchidism will definitely cause infertility, and like I said earlier, it presents a high risk for testicular cancer as well. The ovaries also descend from the abdominal cavity, but only as far as the pelvic cavity. The female gubernaculum extends from the inferior aspect of the ovary to the labioscrotal fold, helping the ovaries reach their final destination. Each gubernaculum becomes a pair of ligaments called the ovarian and round ligaments that ultimately function to support the ovary and uterus, respectively.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.
Okay, that will do it for this episode and the start of the reproductive systems, which is going to be a marathon, not a sprint. We'll be looking at a handful of episodes to cover it all. I want to send out a special thank you to Dr. Kevin Patton for joining us today. Don't forget that I have links to his podcast, website, the MS Happy program at Northeast College of Health Sciences, and HAPS in the show notes. I highly recommend subscribing to his podcast. It is a fantastic listen. And once again, thank you for listening. I hope this show is helping you get your B or better in A&P, or simply helping you understand more about the human body. If you haven't already, please take a minute to rate and review the show on whatever podcast provider you use. And don't forget to tell your friends, classmates, colleagues, and instructors how to find it. Good luck, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. Don't forget to check out my Instagram account and my YouTube channel, both called Student Help for AP. I'm posting a lot of stuff on there right now that you can watch and that will hopefully really help you understand this A&P stuff. Very special thank you out to my family, Bucks County Community College, and McGraw-Hill Education, where you can find my tutor video-based textbook alternative Anatomy and Physiology digital suite, already being used by several colleges and universities throughout the United States.
Your kids could get free or low-cost health coverage from Medicaid or CHIP. Even if you've applied before, they may be eligible now. Kids up to age 19 are covered for checkups, vaccines, dentist visits, hospital care, and more. And if they already have Medicaid or CHIP, remember to renew every year. Visit insurekidsnow.gov or call 877-KIDS-NOW. Paid for by the U.S. Department of Health and Human Services.